Wayne Orr. I'm director for the Center of Cardiovascular Diseases and Sciences at LSU Health Shreveport and vice chair for the Irvin H. Page Committee. The Irvin H. Page Award is one of the premier awards that the ATDB Council gives to junior faculty within their first 5 years of a faculty appointment. In addition to the science, this award is also about the applicant's commitment to the missions of the AHA. So with that, we have the 2024 winner of the Irvin H. Page Junior Faculty Award, Bishuang Cai. So Bishuang, can you tell us a little bit about where you're from, your background, and what the AHA has meant to your career?
I am originally from China, and I came to the United States for my PhD study in 2008. And I did my PhD with Dr. Steve Kaplan at the University of Nebraska Medical Center, where I studied endocytic membrane trafficking. And after I finished my PhD, I joined Dr. Ira Taba's lab at Columbia University Medical Center, where I study cardiometabolic disease, particularly atherosclerosis and metabolic dysfunction associated steatohepatitis.
Yeah. So, what really sparked your interest in metabolic disease?
Yeah, so because I think the prevalence of the obese people is keeping high. So, and there are a lot of complications with associated with these obesities, including diabetes and fatty liver and cardiovascular disease. So I want to study something very fundamental regarding the cellular and molecular mechanisms that drive those disorders.
So the project you spoke on was on this protein EHBP1,
yes.
Right? So tell us a little bit about what that protein does.
Okay, so EHBP1 is the endosomal protein. It helps to traffic or transport cargoes inside the cells. By this way, the cells can communicate with their neighbor. So this particular protein, I'm interested in how it regulates cholesterol metabolism, because it's well known membrane trafficking involved in cholesterol metabolism. Yeah.
So you showed a pretty striking phenotype for EHBP1 modulation, either knockdown or overexpression on hypercholesterolemia. Can you talk a little bit about how that occurs?
Yeah, so based on our study, we found this EHBP1 can inhibit LDL cholesterol clearance, meaning it slows down the uptake of LDL cholesterol from your circulation into your liver. So yeah, when you inhibit it, there will be, when this protein get activated, there will be less LDL get clearance from your blood. That's why yeah.
So with PCSK9 inhibitors really gaining popularity, you showed a pretty strong effect on your protein in regulating PCSK9. Can you talk a bit about that?
Yeah, so the mechanism regarding the action of EHBP1, I think, is through regulating PCSK9 secretion. Yeah, so I think our study is very interesting. We combine two cardiovascular risk genes together, and it seems it's cool.
And you saw a pretty striking phenotype on MASH pathogenesis. Can you tell us a little bit about MASH and how your system is regulating that?
Oh yeah, so usually we think MASH is a risk factor for cardiovascular disease. And in my study, I think it's not like it's not a typical way we think, because my protein seems that it's beneficial for liver, but it may be detrimental for the heart. So we really need to see the fundamental things, how this protein can fine tune to regulate these two diseases in two different ways.
Right. And you showed that the effect on PCSK9, where you were able to blunt PCSK9 expression, that actually caused a worsening of MASH pathogenesis. Do the inhibitors of PCSK9 or statins similarly affect MASH?
Yeah, so we haven't do this kind of experiment in our own system, but according to the literature, I think there is no solid data regarding the effect of statin or PCSK9 inhibitor on MASH patients. So far, I think the result is kind of mixed. So I think we need more study to figure out.
I'd like to congratulate you on joining such a prestigious group of cardiovascular scientists.
Thank you so much.